I will show you the method I use for my heavy metal determinations. Initially, soil samples were accurately weighed and placed into clean digestion tubes, which were initially soaked in 10% nitric acid for 24 hours and subsequently rinsed thoroughly with deionized water to eliminate any potential contaminants. Prior to proceedings, safety precautions were strictly observed, including the use of safety nose mask and gloves to prevent exposure to the corrosive acids. Now, in the next stage of the experiment, the crucial step of preparing aqua regia was undertaken. Now, this was achieved by carefully combining hydrochloric acid and nitric acid in the ratio of 1:3. That means nitric acid is 1 while hydrochloric acid is 3. So for this, you place 3 different beakers. The first one will be for the mixture, the second one will be for the nitric acid, while the other one for the hydrochloric acid. So at this point now, I've measured 450 ml of hydrochloric acid into the glass beaker. Then measured 150 ml of nitric acid. Now note that the quantity of the acid digestion solution added to the samples was tailored to the specific weights of the sample and followed by the established procedures. So this is my procedure. So your own procedure, depending on the amount of or the weight of the soil used, you will now determine the amount of nitric acid to use and hydrochloric acid to use. Now at this point, I pour the hydrochloric acid into the clean beaker. Then I will now pour the nitric acid into the beaker containing the hydrochloric acid. With this, I have succeeded in preparing my aqua regia solution. Now, following this, the prepared acid digestion solution was meticulously added to the soil samples. The amount of acid used depends on the weight of the soil, like I said before, so you have to adhere to your established procedure. Subsequently, the prepared samples were subjected to digestion using a heating apparatus. Here I use the digestion plates, or you call it the digestion digestion plate or the digestion block. The temperature was maintained at 120 degrees Celsius with regular checks for every 2 hours. This is just to monitor the progress of the digestion until a clearer and complete digestion is achieved. So upon completion of the digestion process, the excess acid was carefully evaporated until the liquid volume reduced to less than 1 mil. At this point, the solution was appropriately diluted by a factor of 10 following the prescribed procedure I'm using. The final step involves filtration through a 0.22 micron filter membrane. But before then, you have to prepare your standards. In our next video, I'm going to show you how to prepare your standards. Then after preparing your standards, your samples are ready for the instrumentation. So you use the ICP MS machine to determine your heavy metals. I will also make a video on how the ICP MS works.